Bismillah Rahman Rahim. Welcome again, guys. Welcome to another video lecture by Biology. Today in this video lecture, we are going to talk about elongation process in DNA replication. Okay. In the previous video lecture, if you guys remember, we talked about the DNA replication. We talked the basics of the DNA replication inside the prokaryotes, and we also talked the proteins which are involved in DNA replication. And we also talked about the initiation process in DNA replication in detail. Now, to explain the elongation process in this video, you need to understand the initiation as well as the protein which are involved in DNA replication. Then this video will be very easy for you. Okay, so let me erase this one portion and let me start. Me right? Now, how the elongation process are started in E. coli cell. First of all, let me tell you that what is actually the elongation. Elongation is that stage in DNA replication when a new nucleotides are binded with each other. Okay, whenever the stretch of the nucleotide are binded with each other one and after another and make a long polymer chain of the DNA molecule, we call that what? We call that elongation. And they are continuously to form the DNA chain. Continuously or discontinuously they can make the DNA strand. We call that elongation process. And they can edit the nucleotide until they reach the sequence which we call it ter sequences. We call that what? Elongation process. Okay, now how the elongation are happening in DNA replication. For the elongation, we need to understand that what kind of enzymes which are usually involved in elongation. In elongation, we usually need a enzyme which we call that DNA polymerase 3. Now this is very important enzyme which are used in elongation process. Okay, now let me draw a simple structure of DNA polymerase, and then we will explain that how actually the elongation happen. DNA polymerase have two subunit which we call that beta clamp and clamp loader. So they have usually two subunit. So DNA polymerase will be look like in this form. Beta clamp, they have also alpha sigma subunit as well, but let's suppose we will just see the simple structure. DNA polymerase have very complex structure. They contain alpha, gamma, sigma, all these subunits are connected, but here just I draw the simple structure of the DNA polymerase 3 having two subunit. DNA polymerase 3 can add the nucleotide in 5' to 3' direction. 3' direction. DNA polymerase can start their process. They can start the elongation process by needing a primer which we called as RNA primer. RNA primer, which contain 5 to 10 nucleotide of the RNA. Okay, not the DNA, they contain 5 to 10 nucleotides of the RNA. Okay, so DNA polymerase can start their process whenever they find the primer. Then they can start the elongation process. Okay, that's very important. This primer are provided by an enzyme which we called it DNA primase. We also called this enzyme DNA G protein. DNA G, or we called it primase. Okay. And we have another enzyme which are also involved in this elongation process. We call that DNA polymerase 1. We also need another enzyme, we call that DNA ligase. Ligase. And the last enzyme, not the last, but and we have another enzyme which we call it ribonuclease H, or we also called it RNase. Now, what are the job of the ribonuclease? A ribonuclease can remove the RNA primers, or they can remove the RNA sequences. So we also called it ribonuclease. We also call that what? RNase enzyme. RNase H. These are actually the enzymes. Not these enzymes are used, but we have some other enzyme as well, which are used in the elongation process. Now, how the elongation process are happening? For the elongation, we have, the strand, as we already talked that in initiation, the strand are open. Okay. So let's say again, let me draw the strand here. Okay, so in elongation, we have the structure of the DNA chromosome, bacterial chromosome DNA like that. This is called theta-like structure. Okay, so we get bacterial theta-like structure, just like that. Just like that. Now this is the theta structure. So what actually happened? For the elongation, we already discussed that there is, helicase enzyme, or we call it DNA B protein in this case, and they contain the DNA C protein, which can load this DNA helicase, and this DNA helicase can break the hydrogen bond. Okay, now here is another enzyme that is bounded here. We call that what? We call that DNA gyrase, or we also call it topoisomerase. They can release the tension, or they can, relax the DNA strand, and they can prevent the strand from the supercoiling. Okay, and we have another enzymes which can aid the primer. We call it DNA G or DNA primase. So here we have what? We have the primers. So these are the DNA G protein that can aid the primer, and these are actually the RNA primers. Okay, so we have also the RNA primers added at that specific area. Now what actually happened? These are the, let's suppose these are the enzyme which add the RNA primer. We call that enzyme DNA G protein. Now then DNA polymerase 3 protein are activated, and they can bind there. But here is one problem that are usually created, and the problem is that this upper strand is called as a template strand. Upper strand is the template, and the lower strand is the non-template strand. Okay, now this is also the parental strand as well. Now what actually happened? The primase can bind the RNA primer, then the DNA polymerase 3 are activated. They can bind there at that specific point, but still, as I told you, DNA polymerase can add the nucleotide at 5' to 3' direction, and they can start the elongation process whenever there is RNA primer present. At that specific area, we have the RNA primer. So DNA polymerase can bind here, and they can start the elongation process in 5' to 3' direction. So this is the strand which is going on in 5' to 3' direction. Sorry. 5' to 3' direction. So the strand is the DNA polymerase can move in this way, which we call that 5' to 3' direction. But at lower strand, we have the synthesis of the we have the synthesis of the nucleotide, we have the synthesis, not the synthesis of the, we have the synthesis of the DNA strand, that is usually to 3 prime to 5 prime. Okay, have 3 prime to 5 prime. In this case, what actually happened, we have one DNA polymerase which is used at that specific area, and one DNA polymerase which is used at that specific area. We have one DNA polymerase which is present at that specific area and which is present at that specific area as well. And they can take the two strand and start the elongation process. But the lower strand can move, the lower strand can produce the strand in this way direction, while the upper strand can elongate the strand in this way direction. So here is one question arise, that how the same strand can move in the same direction, because the DNA polymerase can add the nucleotide in 5 prime to 3 prime direction. Then how this one strand will have also moved in this one way? How? We have only one DNA polymerase and they can elongate the strand in one direction, and that is the 5 prime to 3 prime direction. But the lower strand is moving into 3 prime to 5 prime direction. That's the question. That's the main confusion that are usually happen in the elongation process. So how we can solve that phenomena? Now what actually happen? At that specific area we have many nucleotides are, not nucleotides, sorry, we have many primers are attaches. So many of the primers at that specific area we have attaches. But here is only one primer and DNA polymerase can elongate it. So this is another strand which move at that direction. And this strand can move at that direction. So what actually happened? The DNA polymerase that can move in this way direction. So this strand can make a loop. Okay. So whenever the strand can make the loop, so then the DNA polymerase can elongate this one strand as well in this one direction. In this direction, they can also, elongate this strand. This, I am talking about this new one strand, okay? I'm not talking about the old strand. Old strand is already made. This new strand can synthesize in this way direction, while the old strand, while this upper strand can synthesize in this way direction. So when the loop is formed, so then both strands are synthesized in this way direction. But still, the one strand can move in this way direction. 3 prime to 5 prime direction. Okay. So what actually happened? Now, for the formation of the loop, this lower strand can be, unwind so many times as compared to the upper strand. The upper strand is not too much open. So the lower strand can open too much. The lower strand can open. The helicase can perform their function fastly. DNA agarase can perform their function fastly. Everything is going on very fastly. Okay. Then we get a long bubble. Due to this bubble, the strand can be, make a loop. So this strand can be make a loop. So we get the strand like that form. In this form. So this is the formation of the loop. Okay. As I already tell you, there is not only one replication bubble. There is two replication. One replication bubble at that specific area, one replication bubble at that specific area. So one loop formed at that specific area at the lower strand, and one loop is formed at that specific area at the lower strand. Okay, here I just draw one replication bubble, but usually we have two replication bubble. One replication bubble can move at this in this way, which we call it clockwise, and the other can move in this way, which we call that anti-clockwise. Okay, and they can start the elongation process. Now whenever the loop is formed, okay, now this is the strand which is the 5 prime to 3 prime direction. 5 prime to 3 prime direction. 5 prime to 3 prime direction. So remember, whenever we have the nucleotides which are attaches at that specific area, this is the 5 prime to 3 prime direction. 5 prime to 3, and whenever the loop are formed, so then the direction are changed. Okay, then the direction are changed, and this is the 5 prime to 3 prime direction. Direction are now changed. Again we have what? We have the, let me draw again. We have the nucleotides. These are actually the RNA primers. And now the RNA primer direction as looped or formed, so the direction is also changed. Okay, so then DNA polymerase 3 can bind here. So the DNA polymerase 3 can load it at that specific area. And let me draw the DNA polymerase with this red marker. Okay, so this is DNA polymerase 3. Okay, now DNA polymerase 3 can bind at that specific area and they can start the elongation process. But remember, there is no problem at the upper strand. The upper strand can, they just need only one primer, so they can start the addition of the nucleotide and start the elongation process in 5 prime to 3 prime without any issue, without any problem. They can add the nucleotide continuously. So the nucleotide are added here continuously, and we call that continuous strand. This upper strand is called as what? Continuous strand. But at the lower strand, as I already told you that the lower strand are synthesized in 3 prime to 5 prime direction. So what actually happened? We have many of the RNA primers. So these segments are actually RNA primers, and segments, or we can say fragments. These are the RNA fragments, and this RNA fragment was first of all discovered by Okazaki. That's why these fragments are also known as Okazaki fragments. Okay, so we have many of the Okazaki fragments are attached here by this DNA primase. So many of the primers are added here, many primers. Now this DNA polymerase can synthesize the strain into 5 prime to 3 prime direction, and then they reach, this is also the RNA primer, this is also the RNA primer, and then they can synthesize the primers, they can synthesize the nucleotide at this direction, and then they leave it. Now remember what actually happened. This strand is continuously formed. This strand is continuously formed. There's no issue. Okay, at this area they can hold the RNA primer and can add the nucleotide. So we have this. So we have, the nucleotide are added, and they can add at that RNA primer. So when the DNA polymerase reach to that primer, so then they leave this strand. Again, they hold this primer at that specific point and add the nucleotide, and then they leave. Again, they hold this RNA primer at that specific area and start adding nucleotide, and then they leave. Then they hold the strand from that specific area and start the elongation and leave, and the loop are forming so many times. Again and again the loop are formed because we need the same direction. So the loop formation is happening so many times, and the DNA strands are opening, the lower strand are opening so many times. So that's how the elongation are happening so fastly, so quickly. So we can say DNA replication in bacterial cell are, they are very quickly, very fastly the DNA replication are happening in this case. Okay, so this is how the elongation process are continuously started in this one direction. So one replication bubbles moves in this one direction, and the other replication bubbles moves in this one direction, and they can start the elongation process. One DNA polymerase can use at that direction, one DNA polymerase can move in this way direction, but the direction is the same, and that direction is the 5 prime to 3 prime direction. The lower strand can also move in 5 prime to 3 the lower strand can synthesize in 3 prime to 5 prime direction, but as the DNA polymerase can move in 5' to 3' direction. So then what actually happens, the lower strand can make the loop. Due to this loop, the direction are the same. So then they can, when the direction are the same, so then the DNA polymerase can move in this way. DNA polymerase can move, and whenever they are moving, so then the strand are also synthesized. But at the lower strand, they can hold this one primer and add the nucleotide, then leave it. Then they can hold the primer at that specific point and then add the nucleotide, then leave it. Then hold the nucleotide. They usually hold the nucleotide from the 5' area and then leave it. Then hold the nucleotide from 5' area and start the nucleotide adding in 3' direction, then leave it. Then they can add the nucleotide from, there is the 3', so they add the nucleotide into 5' area. Then 5' area, then, and going on, and whenever the loop are formed, so then the nucleotide are added into 5' to 3' direction. But actually it is, the strand is actually what? The strand is actually moving, although it is moving in the same direction, and the primer is also added in the same direction. But actually the lower strand will be, whenever the elongation process are completed, so then you will check, then you will see that actually the lower strand can be, synthesized into 3' to 5' direction. Okay, so the elongation process are started. Then what actually happen, once the elongation process, continues, so then what actually happen, we need to remove these primers, because these are the RNA sequences. We need to remove this primer. Now how we can remove this primer? By adding the, we can remove, not actually we, but it is actually the natural process. The primers are removed by this ribonuclease enzyme or RNase enzyme. RNase enzyme can bind here. This is the RNA or the DNA G protein. Bind here and they can remove the primer. Now the primer are removed out. Many primers are removed. Not primers, sorry, the RNA sequences are removed out with the primers. So whenever more RNA sequences are removed out from the primer, so still we get a gaps. So we have a lot of gaps. These gaps are filled by another enzyme which we call the DNA polymerase 1. Let's suppose the primers are removed at that specific area, at that specific area, at that area, as well as at that area. The primers are removed. DNA polymerase 1 can bind here and they can add the nucleotide. They can add the correct nucleotide. Okay. So then a correct nucleotides will be added. So whenever the correct nucleotide it is added, but remember still the nick are, let me draw here. This is, let's suppose, the RNA primer. So this will be the DNA strand. And again this is the DNA strand. So this RNA primer is removed. This is the template strand, let's suppose. Okay. This RNA primer will be removed by this ribonuclease, so they can remove the primers. So not primers, they can remove the RNA sequences. Now whenever the RNA sequences are removed, so then DNA polymerase can edit the nucleotide here. But still the nick are present. DNA polymerase don't have the ability to seal this nick. They're just adding the nucleotide, but still the bond which is formed between these fragments are still needed. Now how these bonds are joined between these two fragments? Then we have another enzyme which we call it DNA ligase. DNA ligase can bind here and they can seal this nick portion. Okay, so then the nick are sealed by the DNA ligase. And the process is going on. Whenever the nick are produced, DNA ligase can seal it. Whenever the RNA sequences are present, this ribonuclease can remove it. When the DNA ligase ribonuclease can remove it, then DNA polymerase can add the new nucleotide. And the process is going on so many times, and it is very quickly. The process is going on very quickly, and then the strand is elongated and elongated, and this upper strand can be, just jump like that. So if, let's suppose, if whenever this is the new old strand, whenever the new strands are formed, this is the new strand that is formed. So what happen after that? After that, this new strand will be jump like that. They will be jump like that. So we get the strand like in this form. So now the strand jumped, and this is the new strand which is formed. So we have just like in this form direction. Okay, so the elongation are completed. Then at that specific area the elongation is completed and then stopped, and that specific area we call that ter sequences. Ter sequences. There is a ter sequences as well as TUS protein complex are also present. Okay, due to which the elongation process are completed. Okay, so remember, this upper strand can synthesize without any problem, and that's why we call it continuous strand. We also call that leading strand. Leading strand. While the lower strand can synthesize in a segment form. They are synthesized after one segment and then another segment and then another segment, or we can say when the one fragment are synthesized, then another fragments are also synthesized, and then another fragment is synthesized. That's we call that discontinuous strand. That's why we call that discontinuous strand. New strand, or we also call that leading strand. Okay, so that's all about the elongation. The elongation process are started. Let's suppose this is the one replication bubble, and let's suppose this is another replication bubble, and at that specific area the enzyme is present, at that specific area the DNA polymerase is present, and they can start the elongation process, and two replication bubble are moving at a specific point. Means whenever they reach at that specific point, which we call that ter area, ter sequence, ter site, or okay, whenever they reach two replication bubbles, when they reach a ter site, so then the replication, the elongation process will be stopped. Okay, here is not only ter sequences, here is also a TUS protein. So that's all about the elongation. Inshallah, in the next video lecture, when the elongation is completed, when the two strands are completed, two new strands when the new strands synthesis are completed, so how it will be removed, how it will be segregated from each other, we will see it in the termination process. We will talk about TUS sequences, we will talk about the TUS protein complex, and we will see that how actually the DNA strand are finally removed from each other, and how actually how actually the DNA strand are removed from each other, and how the DNA replication are stopped, and how the replication are done in the termination stage. Okay, so I hope this video will be helpful, and I hope you get the idea about the elongation. Just problem is that specific position, so try to memorize it, but still if you have problems, so you can write it into a comment. I will try my best to solve your problem as soon as possible. So if you like this video, so make sure to hit the like button, share this video, and also subscribe the channel, and stay tuned and keep watching the termination video also. Thank you so much.